Good morning, co-chairs of the Global Young Scientists Summit Organizing Committee, esteemed speakers, young scientists and invited guests. Welcome to the Global Young Scientists Summit at One North 2021, or GYSS 2021 in short. I am Kaylee from the National Research Foundation, or NRF, and I will be your MC for today. GYSS is a multidisciplinary summit covering the natural sciences of chemistry, physics and medicine, as well as mathematics, computer science and engineering. This year, for the first time, we are hosting you virtually due to the COVID-19 pandemic. While we are unable to meet and greet you personally, we hope that you will nonetheless be able to feel our warm welcome. I would like to take a minute or two to help you orientate yourself to the GYSS Hop-In platform. Right now, you are on the Stage tab, where all the plenary lectures will be taking place. If you click on the Sessions tab, that will be where you can access the panel discussions and small group discussions if you have registered for any. We encourage you to visit the Networking tab to meet with your like-minded peers and have meaningful interactions. Every day, at 5 p.m. Singapore time, the Expo booths will come alive with our partners who will be sharing more on their programs and initiatives with you. Today, for example, we will have 2 booths going live: NRF's Campus for Research Excellence and Technological Enterprise booth, as well as the NRF Fellowship booth. Do visit to learn more. The Expo tab also hosts our GYSS Help Desk booth. If you require any technical assistance throughout, do click on that and one of us will get back to you shortly. To see the full lineup of lectures and discussions for the whole week, you can click back on the Reception tab and scroll down to see the daily schedule. I hope the brief introduction to the GYSS Hop-In platform will help you navigate the site smoothly. Now, going back to our lineup of events. In the week ahead, we have many exciting lectures and discussions that will provide you opportunities to learn from and interact with the speakers and your peers. To give you a better idea of what's in store, we have prepared a short introductory video. Please sit back and enjoy.
Make sure that our young people are excited about science.
In our research, we tend to get narrower and narrower and narrower as we investigate a problem in more detail. So the importance of conferences for us is that they open our eyes to other possibilities.
Any event which is putting forward the young people is good.
I think that that's wonderful because you guys are our future.
Curiosity, I think, is the most important, and also what is called passion. We really love to understand, not only have to understand, but love.
Knowledge that it was very actual 20 years ago is not actual now anymore, and people need to learn new stuff all the time. So we are in this process of lifelong learning.
Having a forum like this where you get a chance to show young people these great opportunities and share some of the excitement of what you do, hopefully get them to work on problems that you think are important. So I really appreciate the opportunity to do that.
We hope that that short video featuring many of these year's speakers has given you a good idea of what GYSS is about. We now have the honor of having Mr Heng Swee Keat, Deputy Prime Minister of Singapore and Chairman of NRF, deliver this year's opening address. Here is his pre-recorded speech. Please enjoy.
Welcome to the Global Young Scientists Summit 2021. The 9th edition of this summit takes place in the shadows of COVID-19. The pandemic has infected more than 80 million people and taken nearly 2 million lives. But we start the year with a ray of hope. Several vaccines have been authorized for use, and vaccination is underway in many parts of the world. However, the work is far from over. The vaccination of a significant proportion of the global population will take time and resources, and the emergence of a new, more infectious strain of the virus is a reminder that we must be prepared for surprises ahead. What is certain is the critical role of the global scientific community in the fight against COVID-19. Early in the crisis, the sharing of the virus genome with the global community enabled the rapid development of diagnostic test kits. Your collective efforts in the past year has led to the development of safe and effective vaccines in record time. Your many other innovations have helped businesses, workers, and households adapt to the crisis. Your continued research and innovations will be crucial in helping us overcome the pandemic and build back better from this crisis. While each of us can pursue research excellence individually, our efforts are greatly multiplied if we work together. As French biologist Louis Pasteur once said, "Science knows no country because knowledge belongs to humanity and is the torch which illuminates the world." We must build on the positive momentum of collaboration in the fight against COVID-19 and redouble our efforts to work in stronger partnership across disciplines, across borders, and across industries, academia, and governments. This will allow us to not only overcome this pandemic, but also other global challenges such as poverty, aging, and climate change. Where I stand now, the Singapore ETH Centre is a collaboration between ETH Zurich in Switzerland and our National Research Foundation. This centre is part of CREATE. CREATE stands for Campus for Research Excellence and Technological Enterprise. The campus is home to other research collaborations set up with leading global universities and research institutes, including CNRS from France, TUM from Germany, MIT from the US, and Shanghai Jiaotong from China. Together, we work on cutting-edge research to tackle global challenges. An example of such a program is Cooling Singapore, a project to reduce urban heat in Singapore. This is an interdisciplinary and multi-institutional initiative between Singapore-based scientists and several of our international partners based at CREATE. The solutions developed will certainly be useful for Singapore, but can also contribute to reducing urban heat in other cities in the world. As we strengthen global partnerships, we are also deepening Singapore's R&D capabilities. Over the decades, we have built up deep research capabilities in areas such as quantum technologies, immunology, and photonics. In quantum technologies, we established the Centre for Quantum Technologies at the National University of Singapore. This is one of the international reference research centres in quantum information science. We developed the world's first quantum nanosatellite that can generate entangled photon pairs in space, which is a step towards quantum secure communications. In immunology, we have developed cutting-edge capabilities in CAR T-cell therapy, where we have developed and administered pioneering treatments for patients with acute lymphoblastic leukemia at the National University Hospital. In photonics, we have a strong R&D base spanning basic and applied research at A-STAR and our universities, including the Nanyang Technological University, allowing us to be the world's first to demonstrate the control of light rays through switchable nano antennas. This paved the way for smart devices to deliver high-resolution 3D moving images. I encourage you to visit the expo area of this summit to learn more about the many possibilities for collaboration from the exhibitors. In the next 5 years, Singapore will also be investing $25 billion in research, innovation, and enterprise. Our commitment to research over the decades remains steadfast, despite the economic cycles and headwinds. By working in collaboration with partners from around the world, we will tackle a wider range of strategic priorities and emerging opportunities. This will enable us to contribute to societal and global advancements in areas such as human potential, trade and connectivity, environmental sustainability, and digital innovation. We'll continue to stay true to our vision to be a knowledge-based, innovation-driven nation, fueled by scientific excellence. With the summit being held online this year, we're expecting more than 500 aspiring researchers and scientists from across 85 institutions in 30 countries. We also welcome 21 plenary speakers and panelists who are distinguished leaders in their respective fields. Among them are Nobel laureates, field medalists, Turing Award, and Millennium Technology Prize winners. They all share a passion for science and discovery, a dedication to the frontiers of innovation, and a desire to share their knowledge with others. Professor Robert Langer will be leading the plenary session. Moderna, a company he co-founded, successfully developed a COVID-19 vaccine. His reflections on the development of drug delivery systems will be a fitting start to the summit. I thank our speakers and panelists for taking part in the summit, and I hope that you will be inspired by them. When air travel resumes, I welcome you to visit Singapore. We look forward to working with you and hope that you will spend time with us to further your research endeavors. We also have various competitive fellowships and investigatorship programs to support you in the pursuit of groundbreaking research in Singapore. By working together, we can build back better from this crisis and create a better and brighter future for the world. I wish you all a fruitful summit. Thank you.
And that brings us to the end of the opening ceremony. We hope you will take the opportunity to start networking and build friendships with your fellow scientists. I would also like to take this opportunity to ask you to follow us on social media. Find us by searching for National Research Foundation Singapore on Facebook and Instagram, and GYSS_SG on Twitter. The opening plenary session by Professor Robert Langer will commence shortly, so please stay on the stage for that. Following that will be a panel discussion on personalized medicine featuring Professor Thomas Cech and Professor Jennifer Doudna, who recently was awarded the Nobel Prize for Chemistry. In the second part of today's program, we also have two plenary lectures, the first by Professor Aaron Chekanova and the second by Professor Ada Yunath, as well as a panel discussion on science and policy by Dr. Venki Ramakrishnan and Professor Tan Chor Chuan. So that's a full day of program lineups. Once again, thank you for joining us and being a part of GYSS 2021. We wish you a fruitful summit.
Okay. Okay. We can start. Good morning. This is Subra Suresh, President of Nanyang Technological University in Singapore. It's a great pleasure and privilege to introduce our plenary speaker this morning, Professor Robert Langer from the Massachusetts Institute of Technology. Professor Langer is a longtime friend and colleague with whom I've had the distinct pleasure of collaborating for many years. Let me take a few minutes to give an introduction. Let me take a few minutes to give an introduction to his. You can continue. Professor Langer is one of 11 institute professors at the Massachusetts Institute of Technology. He has written over 1,500 articles, which have been cited over 320,000 times. He has more than 1,400 issued and pending patents worldwide. His patents have been licensed or sub-licensed to over 400 companies. He has served as chairman of the Federal Drug Administration in the US Science Board, its highest advisory board, from 1999 to 2002. His awards, which are over 220, are too numerous to list here. I will just mention a few. He has received both the US National Medal of Science and the National Medal of Technology and Innovation. The Charles Stark Draper Prize, which is the Engineering Nobel Prize, equivalent to the Engineering Nobel Prize given by the National Academy of Engineering in Washington, D.C. The Queen Elizabeth Prize for Engineering, the Breakthrough Prize in Life Sciences, the Kyoto Prize, the Millennium Technology Prize, and many, many others. The Lemelson MIT Prize recognized him for being one of history's most prolific inventors in medicine. He holds 34 honorary doctorate degrees, and he is one of very few Americans elected to all three branches of the US National Academies: the National Academy of Engineering, the National Academy of Sciences, and the National Academy of Medicine. In addition to that, he has been elected to the American Academy of Arts and Sciences and the National Academy of Inventors. So it's my great pleasure to welcome my good friend, Professor Robert Langer. The title of the plenary talk is The Edison of Medicine: Robert Langer's Quest to Solve Global Health Challenges Using Biotechnology. Bob.
Thank you. Thanks so much. Thank you.
To be able to speak with you. I'd like to start by telling you a little bit about my career when I was the age of many of you. When I was like a lot about your old age, I was a graduate student at MIT in chemical engineering. And when I got done with my doctoral degree, pretty much all my friends, this was 1974, went into work for oil companies. That's what chemical engineers did. So I thought that's what I should do too, just what my friends did. So in all the oil companies, there was like a gas shortage around then, and they had all these high-paying jobs. So I applied to the oil companies, and I actually got 20 job offers, 4 actually from Exxon alone. And I wasn't that good, but they just had so many openings. One of them made quite an impression on me. I remember going to Exxon in Texas or Louisiana, and when I went there, one of the older engineers said to me, if you could just increase the yield of this one chemical by 0.1%, he said, that would be wonderful. He said it'd be worth billions of dollars. And I remember flying home that night thinking to myself that I really didn't want to do that. And I just thought it would be boring and unimportant. So it made me think about what I did want to do. And when I was a graduate student at MIT, in addition to doing my thesis, I helped start a school for poor high school children. And I got very involved in it, and I helped develop new chemistry curriculum, new math curriculum, to try to make math and science very interesting for people even who had dropped out of school. And so one day I saw an ad to be assistant professor at what's called City College of New York. And it was to do just that, develop new exciting curriculum. So I wrote them a letter applying for a job as an assistant professor, but they didn't write me back. But I really liked that idea, so I found all the ads I could to do something like that. I found about 40, and I wrote to all those colleges, and actually not a single one wrote me back. So that wasn't going so well. So I started to think, what other way could I use my chemical engineering education to help people? So I thought about medicine. So I applied to a lot of hospitals and medical schools. They didn't write me back either. But one day, one of the people in the lab where I was, one of the older people, he said, Bob, he said, there's a surgeon in Boston named Judah Folkman. And he said, sometimes he hires unusual people. He thought very highly of Dr. Folkman. I won't say what he thought about me. But anyhow, I wrote to Dr. Folkman, and he was kind enough to offer me a job as a postdoc at Boston Children's Hospital and Harvard Medical School. And the problem that he wanted me to work on is shown on the next slide. He had this idea that if you could stop blood vessels, maybe you could stop cancer. And if you look at this picture, this was from 1971, actually. On the bottom left, you see a depiction of a tumor cell. Somehow it's become abnormal and it starts growing abnormally. And then as you see the curve going, it grows bigger. It grows to about, if you look in the middle, the lower center of the screen, what you see is it's probably about 1 million cells. And it won't actually get bigger than that because it runs into a nutrition problem, except for one thing. He says that the tumor, he believes, creates a chemical signal called TAF, or tumor angiogenesis factor. And what that does is it causes the surrounding blood vessels, which normally don't do anything, to start growing right to the tumor. And they connect with the tumor, and that solves the nutrition problem for the tumor. And then, as you see on the upper right of this picture, the tumor gets bigger and bigger because it has blood vessels in it. And if you go to the uppermost picture, you see that the tumor is highly vascularized, and actually it spreads through those blood vessels. That's a process called metastasis, which often kills people. So my job, he said, was to prove that he was right. It turned out that this was an incredibly controversial theory. Almost everybody at this time in the early 70s didn't believe it. They said it wasn't right. And he wanted me to prove it, but in so doing, isolate the first substances that could stop blood vessels from growing. So how do you think about a problem like that? Well, we broke it down to 2 parts. First, where could you ever find something that would stop blood vessels? And we thought about cartilage, which is in your nose and your knee, and it doesn't have blood vessels. So I was able to get like little rabbits in the lab, but I couldn't get much cartilage from them. So in chemical engineering, you learn about scale-up. So I went to cows, and I got some cow bones at a local slaughterhouse. But still, getting 1 or 2 bones, you can't get that much from that either. So I tried to find out, where do all the cow bones in the northeastern United States go? And it turns out they go to these meat packing places in South Boston. So I made an arrangement with them to get all their bones. I'd go there 4 times a week. I'd bring their bones back. It totally occupied my entire car. And I bring them back, and I'd scrape the meat off of the bones. Let me just show you a picture on the next slide. And this is a cow bone, and the top of it's cartilage. You see when the meat's scraped off, it's like white and glistening, and there are no blood vessels. So what I do is I slice off that cartilage piece in the top. I'd extract it in solvents like with guanidinium hydrochloride to break the non-covalent bonds, and then I'd put it through different purification procedures. So that at the end of about a couple of years, I had like what I'll call 100 different fractions from the cartilage. And I needed to test them. So that brings up the second problem. How do you test whether you're stopping blood vessels from growing? And what you need to do, and this almost always has been an issue in any type of time in history, when anybody's ever found a new factor, is you have to have what's called a bioassay. And there was no bioassay for studying blood vessels, so we had to invent one. And what Dr. Folkman and I tried to do is shown on the next slide. This was our idea, that maybe you could put a certain type of tumor, we call a V2 carcinoma, in the eye of a rabbit. Rabbits have big eyes, so they're easy to visualize through an ophthalmic microscope. And also the tumor will bring blood vessels in from the edge of the cornea, what's called the limbus, but it takes about 9 weeks. So what you want is something we thought that could be not inflammatory in the eyes, like maybe a polymer or lipid, something like that, so it wouldn't cause any damage. And then it would also have to be able to release these molecules in the eye for whatever period of time we wanted, like say 9 weeks in this case. And the beauty of the eye is that now you could actually use an ophthalmic microscope to measure the length of the longest blood vessels. So Dr. Folkman, he was on the board of the only company in the world that had ever tried to deliver it's called ALSA. They had Nobel laureates in chemistry on their board, and it was a very distinguished group. And he asked them, could they help us? But what they said, next slide, is that you couldn't do it. They basically said, if we go to this next slide, that you can't do that because we wanted to use polymers, and they said the big molecules, they can't get through them. It's like any of us walking through a wall. I mean, you might be able to make it very, very porous, but then it comes out immediately. So the challenge is, could we slowly deliver these molecules for at least a long enough time to do something? In fact, the literature said the same thing. Next slide. And really, I think the only thing I had going for me is I hadn't read any of this. So I tried anyhow. And I spent 2 years working in the laboratory, experimenting with many different techniques, different materials, different designs. And I found over 200 different ways to get this to not work. But eventually I found out a way to make little tiny particles, microspheres, nanospheres. And let me just show you a picture on the next slide. Here you see on the left is a picture of one of these, and on the right it's cut in half. And what we did, next slide, is we published in the journal Nature in 1976 that you could use this to release anything, any protein, and actually this was also, it turns out, and this will come into my talk later, it's actually the very first time anybody developed a delivery system for nucleic acids like RNA or DNA. So, so that was very exciting to me that we could deliver all these molecules. In fact, we even, as we went further, worked out ways to release them constantly if we wanted to. And a picture of that is shown in the next slide. So I was all very excited about that. But then in 1976, around the time of that paper, I was asked to give a big speech at a major scientific conference in Michigan. And I was 27. I was the youngest person there. And I practiced this talk for weeks. And finally, I got up, and and there were all these very distinguished scientists at the meeting, and who were experts in materials and engineering and chemistry. I got up and I gave that talk. It was a 20-minute talk, and at the end of it, I actually thought I did all right. I didn't forget too much what I was going to say. I didn't stammer too much. So I thought when I was done with this talk, all these older scientists, being nice people, would want to encourage me, this young guy. But when I got done, I stepped off the podium, and a whole bunch of them came up to me, and they said, "We don't believe anything you just said." They said, next slide, that the large molecules are just too big to get through. And we were also using organic solvents, and they said that will denature whatever you put in, peptides, proteins, nucleic acids. So they said this has got to be wrong. And actually, because of all that, things went downhill from there. Actually, I tried to get research grants. My first 9 grants were rejected. And then a lot of my friends said I should try to get a job, not just be a postdoc. So, I listened to them, and I'm a chemical engineer, like I said, so I applied for a lot of faculty positions in chemical engineering departments, but not a single chemical engineering department in the world would hire me. So I didn't get any jobs there. Finally, Dr. Folkman, he knew a man named Nevin Scrimshaw, who was head of the nutrition department at MIT, and he introduced me to him, and Dr. Scrimshaw was kind enough to offer me a job. But the only thing is, Dr. Scrimshaw was a great man, but he was what I'd almost call like a benevolent dictator. He liked me, so he hired me, but he didn't ask anybody else in the department what they thought. That probably would have been okay, except for one thing. And that is, the year after I joined the department, Dr. Scrimshaw left. So a lot of the senior faculty decided to give me advice, and their advice is I should leave too. In fact, on the next slide, I tell you a story. This is from a few years ago, actually. Michael Marletta, he's a very famous chemist. He was head of Berkeley's chemistry department, head of Scripps, member of the U.S. National Academy of Sciences. He was giving a speech when I got an award, and he was saying what it was like to be with me in the late 1970s and early 80s at MIT, and I'll just read this to you. He said one evening he went to a faculty dinner at a Chinese restaurant with me and some senior MIT professors. He said a senior scientist sat quizzing us while smoking a cigar. He said when the older scientist heard my concepts for drug delivery, he blew a cloud of smoke in my face and said, "You better start looking for another job." Mike said he thought he was in a Fellini movie. So that was very discouraging, but I didn't have much choice. I just kept trying to work. Even though nobody believed it, I tried to understand it better. And I had one of my graduate students, Rajan Bawa, cut thin slices through our systems using a cryomicrotome, a low temperature cutting instrument. So on the next slide, let me just show you a picture of one of those. And so if molecules were really big, they couldn't get through this. Now, it turns out when we put like a model molecule in, like here we picked the red protein on the next slide, and then we put it in using the way we made these, it's looked very different. This is a thin section, a 5-micron thin section cut before any release has taken place. And you see what we call a phase separation. Polymer is the white part, and the drug, the myoglobin, so to speak, that's the red part. Now, if you release this, say, for a year, and then you look at it, it looks tremendously different. And that's shown on the next slide. So here, what you see are all these pores that have been formed. They weren't there 2 slides ago. And it turns out that these pores are interconnected. They're also incredibly winding and tortuous. So it takes a very, very long time for the molecules to get through. And they're also, even though they're connected, the pores, there are tight constrictions between them. And so that makes a really long time for the molecule to get through. And it turns out over the years, a lot of my students, like Bill Ryan and Dean Shea and Larry Brown, Mark Saltzman, they've worked out mathematical models to predict exactly how you can control these pore structures, what materials you can use, so you can get almost any release rates you want, from days to years. And I apologize in advance for the slide I'm going to show you now, because it's a little bit graphic. It has blood vessels in it. But there's no other way to illustrate this. So what we're going to do is look at this 9 weeks later. We actually did 2,000 eyes, 100 fractions or so. And I'm only going to show you representative ones from the paper. But this is at 9 weeks. If we could just go to the next slide, what you'll see is on the left-hand side, without the cartilage-derived inhibitor, and this is true for almost every fraction we tested too, because almost every one didn't work. But what you see on the left is a sheet of blood vessels growing over the polymer to the tumor. And in fact, if you looked at this eye 3 weeks later, it would be 3-dimensional. Out of the orbit of the eye, it would kill. But when we put the inhibitor in, or the one thing that turned out to be the inhibitor in, take a look on the right-hand side. And we repeated this 21 times. And you see the vessels are lower, they're sparser, and they actually avoid the polymer. And 60% of the time, these tumors never grew. So that was very exciting, and that showed for the first time that blood vessel inhibitors did exist. And also it provided a way for future people to study this. Now, this is 1976 academic study. It still took another 28 years from our paper and billions of dollars and great companies like Genentech doing work on this before the first blood vessel inhibitor was approved by FDA. And let me just show you what happened starting 28 years later. Next slide. What you see is starting in 2004, 28 years after we started, is one after the next, after the next, after the next blood vessel inhibitor approved by the FDA. Avastin, the first of these, is which was originally approved for colorectal cancer. It's probably the #2 largest selling biotech drug in history. And of course, these have been approved for many, many different diseases, many types of cancer. And not only that, they've been widely used. In fact, they're the only therapy, pharmacologic therapy, for diseases of blindness in the back of the eye, like macular degeneration and diabetic retinopathy. In fact, sometimes they can actually reverse these diseases. So those very historic, and these are used in tens of millions of people every year now. So that showed that you could actually, and now many of these, and this continues to today, have these approvals by FDA. And these drugs are all blockbusters, sell many billions a year. But, you know, I also thought that maybe the drug delivery work we've done could be useful in its own right. So one of my graduate students at MIT, Larry Brown, just as a proof of principle, showed, as we'll see on the next slide, that you could design, and I'm just showing you one graph from his whole thesis, but if we go to the next slide, what you could see is he was able to lower the blood sugar level of a diabetic rat from 400, which is diabetic, to 100, which is normal, for over 100 days. And yet that's with a single implant. And so one day, Dr. Folkman and I, Dr. Folkman said to me, Bob, we should file a patent. And so we did. But 5 years in a row, the patent examiner rejected it. In fact, it's interesting. This was the first patent in the history of Boston Children's Hospital, which is a very good hospital. But we've got rejected 5 years in a row. So the head of the technology transfer office at the hospital came to see me one day. He said, Bob, you have to stop. He said, this examiner's never going to allow this patent. Just forget it. You're wasting an enormous amount of money for the hospital. He said, just give up. But I don't like to give up. So I started to think, how could we convince the patent examiner to allow this patent, legally, of course. And science really wasn't working. But as I told you before, when I first started doing this, everybody told me it was impossible. It could never work. So I wondered, did anybody ever write that down? So you can do what's called a science citation search. I did this in 1982. So remember, the Nature paper we wrote was 1976. So I looked back, see who cited that paper and what they said. And I found hundreds of references. But I thought I'd just show you one. And this is on the next slide. So this has actually turned out to be written in 1979. I had no idea. By the way, it's an interesting exercise to see who writes about what you do. I had never done this before. So, but this was really very helpful. These 5 people wrote that describing the field that the agent to be released is a small molecule with a molecular weight no larger than a few hundred. One would not expect that macromolecules, they said, like proteins, could be released by such a technique because of their extremely small permeation rates. However, Folkman and myself have reported some surprising. So surprising is a really good word to show to a patent examiner. Surprising results that clearly demonstrate the opposite. So I showed that to the head of the tech transfer office. He showed it to the patent examiner. And the patent examiner said, I had no idea. He said, I tell you what, I will allow this patent if Dr. Langer can get written affidavits from these 5 scientists that they really wrote that. So I was this young guy, but I assistant professor. I wrote them all, and they're all nice enough to write me back that they really did write that. And the patent examiner allowed the patent. The next slide shows that. With that, we would start different companies. We would teach different people how to do this, teach different companies how to make these. And what's happened because of that, next slide, is there are many, many products used really probably by hundreds of millions of people all over the world. These are just a few. But there are, like in the middle right of the screen, there's Lupron Depot. That's a little microsphere. if you tried to swallow, luteinizing hormone, releasing hormone analogs, it won't work because they'll get destroyed and they're too big to be absorbed. So they tried to inject it, but they were destroyed that way too. So they put them in these little microspheres, inject them subcutaneously, and they become a widely used therapy for hundreds of millions of patients. And there are many, many others. New ways of treating opioid addiction, pain, cancer, heart disease, different eye diseases, ways to stop bleeding, and mental health diseases like schizophrenia. All these are different ones that have been approved here. Type 2 diabetes. So that was very rewarding to me to see that could happen. Also, one of the things we did is not only, a lot of these that I'm showing you are microspheres that you might inject intramuscularly or subcutaneously, but we also thought we could make nanospheres and inject them into the body to do different things. And as I mentioned to you, the Nature paper was the first time anybody put in large molecules and those as well. So I thought I'd show you, and we also figured out ways. One of the other problems is when you inject nanospheres into the body, they get destroyed right away. And we developed certain types of coating, like what's called polyethylene glycol. It attracts a lot of water. And that coating actually enables the nanospheres to last long enough to do good. And the next slide is going to show you a video that explains this far better than me. I wish I could do as well.
He starts with a nanoparticle of anti-cancer drug. That gets encased in a plastic that releases the drug over time. That, in turn, gets a special wrapping that disguises the package as a water molecule to fool the body's immune system. And last but not least, the address where it should be delivered, a key that will only fit the lock of cancer cells.
Now, I should point out that a lot of the clinicians I work with tell me it doesn't blow the cell up quite like that. But this kind of nanotype approach works on all kinds of things. We use it for small molecules. And then it's actually been very useful for large molecules. So, for example, Alnylam is a local company in the Boston area. And I was asked to be on the scientific advisory board from the beginning. And Alnylam got the very first, I helped them, and they got the very first what's called siRNA approval. And basically they did it by putting it in nanospheres. That's for a very rare disease called ATTR amyloidosis. That's been very exciting. And then in 2010, I myself and one or two other people, we started this company just actually less than a block from myself, which is called Moderna. And the whole idea of Moderna was to develop new drugs of messenger RNA. And messenger RNA can be very, very useful. But again, if you try to inject it into the body, it's destroyed right away. So what we do is we put it in these little nanoparticles, and they, in this case, made of lipids, and basically, but they still have this coating, and basically we inject them in, and they last long enough to make different things. Probably the classic example that many of you have heard about is the vaccine for COVID, and basically we've made a messenger RNA, put it in nanoparticles to make a COVID vaccine, and hopefully that will be approved by regulatory authorities in on the 11th. At the end of this week. So at any rate, all these drug delivery things actually enable many therapies that would never have existed before. By the way, every company, Pfizer, BioNTech, Moderna, CureVac, Novavax, all these companies use nanoparticles to deliver like we originally designed. They've improved them from what we did, but to deliver and protect mRNA. Now, I want to go on and tell you some other things that we created. One of the things that became curious about when I worked in the lab at the hospital was how did materials find their way into medicine? And when I looked into this, I found it was almost always done by medical doctors. And what they do is they'd go to their house and they'd find some object that kind of resembled the organ or tissue they wanted to fix. So it turns out that the artificial heart is a lady's girdle material, so it has a good flex life. And a mattress stuffing is actually used for breast implants because it's squishy. So I thought, well, we could do better. Rather than take materials from your house, what we could do is actually use what I'll call chemical engineering design. Ask the question, what do we really want from a biomaterial, from an engineering standpoint, chemistry standpoint, biology standpoint, and then design it from scratch. So we picked an example. And the example I chose was synthetic degradable polymers. The next slide shows an example of how almost all degradable polymers dissolve. And they dissolve by a process that I'll call bulk erosion. So if you start on the top, it looks like a sheet, but then it gets spongy. And then by the time you get to the bottom, it falls apart. That may be okay for some drugs, but for others, it's as you see, it gets so porous, the drugs could just burst out. So if you had a cancer drug or if you had insulin, that would be very bad. So we said, from an engineering standpoint, we don't want the polymers to dissolve like that. We want them to dissolve like as shown in the next slide. Surface erosion. And we wrote on the blackboard all the criteria that we wanted to do this. So we wanted to make, you can make choices like what should cause the polymer to dissolve, like water or an enzyme. Well, everybody has different enzyme levels, but you all have excess water. So we said, we'll build into the new polymer the fact that water's got to make it degrade. And then we said, if we do that, what should the building blocks be? We need them to be safe and non-toxic. And what should the bonds be? So what we came up with, without going into a huge amount of detail, is shown in the next slide. And it's a polymer. It's called a polyanhydride. And there are two parts to it. It's what's called a copolymer. Sebacic acid and carboxyphenoxypropane. And it's connected by anhydride bonds. But our theory was that if you could change the ratio of these two, you can get almost any erosion rate you want. And that turns out to be true. That's shown on the next slide. And even if you take a thin system, you can make this, here we're on this slide, we're changing the ratio of the two units. 79% of the one, 55% of it, 15%. But if you go to 0%, you can imagine that that'll take about 4 years to dissolve, whereas the 79% one is gone in 2 weeks, and we could make it go faster. So one of the things that's been very important to me, I love doing the basic research. I love doing curiosity-driven research, but I also want to see this help people. So in 1985, a young neurosurgeon named Henry Brem came to see me. And he was then just starting as a neurosurgeon. Now he's actually head of neurosurgery at Johns Hopkins. But he wanted to figure out a better way to treat brain cancer. And the worst and most common form is shown in the next slide, glioblastoma multiforme. And no matter what you do, the mean lifespan is less than a year. And the only drug that was approved at that time for use is shown in the next slide. And this drug is incredibly toxic. You give an injection, and it causes terrible side effects to the liver, kidney, and spleen. So Henry and I had an idea that maybe we could come up with a new paradigm. And that idea is what we'll call local chemotherapy. And this is shown on the next slide, where what the neurosurgeon might do, like Henry, is line the surgical cavity with a polymer that contains BCNU, the drug. Now, normally, it would only last 12 minutes, but when it's in the polymer, it's protected from degradation, and it will last as long as the polymer lasts. And we could make this polymer last any length of time, like a month is what Dr. Brem wanted. So we did that by changing the copolymer ratio. But the power of this is you get high concentrations in the brain where you want it, and low concentrations, this is the fourth bullet, in the brain where you want it to kill the tumor. So high concentrations in the brain where you want it, but low concentrations everywhere else. So when we first proposed this, you have to raise money. And the way we would raise money is by writing grants to the government. And they'd have what are called study sections. These are other professors who review what we do, and they decide whether we should get the money. And I was very excited about this, but our grants did terribly. The next slide gives a brief summary of what happened. In 1981, when we first proposed this, the reviewer said the polymers can't be synthesized. But I had a really good graduate student, Howie Rosen, who later became CEO of a $12 billion company in drug delivery, and he showed they could be synthesized. Then people said, well, okay, but these polymers, they'll react with whatever drug you put in because of these anhydride bonds. But another postdoc, Cam Leung, and he showed, now he's a very distinguished professor of a chair at Columbia University, he showed that didn't happen. So then we sent it back again, and the reviewer said, well, we still shouldn't fund this because these materials are fragile, they'll break in the body, they're low molecular weight. But I had another couple of postdocs, Edith Mathiowicz, who's a full professor now at Brown University, and Avi Dome, who's dean of pharmacy at Hebrew University, and they showed that we could make really strong, high molecular weight polymers. We sent it back again, and the reviewer said, well, we still shouldn't fund this because this will be a new polymer, it's going to be toxic. But Cato Lorenzen, one of my students, who later became the Dean of Medicine at University of Connecticut, and also received the National Medal of Technology from President Obama for this work, he showed that, and other work, showed that it wasn't toxic at all. So then the reviewer said, well, okay, but the drug won't diffuse far enough to kill the remaining tumor. But I had another graduate student, Mark Saltzman, who Yale University asked to be their initial head of biomedical engineering. And he's won all kinds of honors. And he showed it did diffuse far enough. Anyhow, this went on and on until 1996 when the FDA approved it. It was the first time in over 20 years the FDA approved a new treatment for brain cancer, and the first time they ever approved this idea of local chemotherapy. You can probably tell from the way I'm speaking that I'm very proud of how well all the MIT people did. They got great jobs and honors, whereas the reviewers, they haven't done so well. Now let me just show you a little bit about how this works. I'll show you, again, a picture of a brain with a little wafer in it on the next slide. And on the next slide, we'll show you, the following slide, I'll show you how it usually looks. They put several of these in before closing up the brain. Next slide. Next slide. I don't like to leave those on too long. So basically, there have been a number of clinical trials. This was one of the very first ones. And it's not a cure by any means, but it did show that you could prolong life by, in this case, in this study, about 5-fold increase in survival compared to controls, which are conventional best treatments. And even a couple of years ago, they analyzed, the next slide, about 60 clinical trials, again showing that it was statistically significant. Actually, so I should have pointed out that it got approved in 1996. It's still used today. Basically, it's been approved in over 30 countries and used on thousands and thousands of patients for now the last 24 years in these 30 countries. It also created a new paradigm for other treatments, in particular, and this, what I'm going to show you shows how you never know where things will lead. So this is treating cancer. But one of the biggest problems in heart disease, as you probably all know, is you get blockage of blood vessels. So a way that people treat that is what are called by putting stents in. These are like Chinese finger puzzles to prop the blood vessels open. And a picture of that shown on the next slide. But what happens is, as you may know, is about 60% of the time you get the smooth muscle cells proliferating when you put these stents in, and the blood vessels close, and the patient can die. And so, and you have to put a new stent in the best case, but death in the worst case. So what they now do is several companies like Boston Scientific and others, they make these coatings of polymer, and they put a different anti-cancer drug in, even though it's for heart disease. That drug is Taxol, but it's anti-proliferative, and it locally delivers the drug. If you gave it systemically, patient would die. But they give it locally, and it's like 1/1000th the dose, and it keeps the blood vessels open, and millions of these are used every year. So these are two things that I wanted to go over with you on drug delivery, but I want to now turn and end the talk on how you maybe could use materials to someday create new tissues and organs. And here I've worked very closely with Ajay Vakanti, who's a good friend of mine and a surgeon, initially at Boston Children's and now at Mass General. And about 30, almost 40 years ago, he came to see me when he was heading up the liver transplant program at Boston Children's Hospital. He wanted to make a new liver for this little baby on the next slide. And the problem is that child has to get a transplant. If he doesn't, he would die. And so Jay once asked me, he said, well, Bob, could we make new tissues and organs from scratch? So our idea, this is from a paper we wrote in Science, next slide, that's been cited, I think, almost 8,000 times. Next slide. Our idea is we could take almost any cell type, and these are shown on the left. By the way, we could also take stem cells, but people didn't talk about stem cells then. So here we put different cell types in, we dissociate them. If you injected these cells at random, not much would happen. But the cells are smart, and people have shown that if you put cells close enough together, they can recreate tissue structures. And so here what you see is an example of a polymer scaffold that we want to make where we could put the cells close enough together. Then we grow it in a bioreactor in vitro. And then the idea is you could put it in the patient and make virtually any tissue you want. And once again, we created new materials. The next slide shows some examples of that. We either use conventional FDA-approved materials like lactic glycolic acid, and these are degradable, but sometimes cells won't adhere to this. So what we wanted to do was build in the ability for cells to adhere if we wanted to. So we also created new polymers like lactic acid colysine. And notice on the bottom picture, depiction, you see where the lysine is the NH2 group. So for the chemists in the audience, you could use what's called a carbodiamide reaction and link almost any set of amino acids or other things to it. And that's actually helped us in many different tissues we've tried to create. Now, on the next slide, what we've done is we've formed them into fibrous structures. And here, like you see, there's a lot of porosity, and this we put liver cells on. But the beauty of engineering is you can make almost anything. So Prasad Chastri, who was one of my postdocs, he now runs an institute in Germany, actually had this idea of using CAD-CAM techniques. That's computer-aided design. And then you follow it by something like three-dimensional printing or different foaming techniques, and you could make these systems in any shape at almost any porosity. So as an example, he picked a nose. And what I'm going to tell you now is speculation, but let's say 30 or 40 years from now, somebody goes to a plastic surgeon and they don't like their nose. They want a new nose. So our idea was that you would have a computer screen. You could pick your choice of 40 noses. So the next slide shows an example of that, where he's on the left, you see a nose of that doesn't have cells on it, but it's 98% porous. And then you could take the cartilage cells actually from the patient themselves. I'll show you an example of that in a few minutes. And you could make a nose. Now, this is like a regular nose, but if you wanted, say, an upturned nose, you could do that. You could just take a little bit of the cartilage off the spongy one on the left, and maybe somebody would want a hook nose. They probably wouldn't, but if they did, we could make that too. So just to show you a few examples of making cartilage. Next slide. Why don't I show the next slide? So with the Vacantes and Lisa Freed and Gordana Vunyak, we actually took nude mice and took their own cells and made, as you can see in the bottom, we redid his skull. And on the next slide, we redid the top animal's cheek. And if you open them up and look at them, it's pure white glistening cartilage. And I'll show you that on the next slide. And if you actually do what's called histology, that's a way to judge about what you made. What you'll see on the next slide is it looks like almost a textbook of cartilage. The characteristics of cartilage are having glycosaminoglycans, of having type 2 collagen, of staining with saffron and O, and having these chondrocytes in characteristic lacunae, which you see on the bottom two parts, C and D. And finally, staining for chondroitin sulfate. That's very characteristic of cartilage, and that's with an Alcian blue stain. So it looks just like cartilage. The only thing is it's not as good as regular cartilage. If any of you are runners and thinking we can help you, we're not there yet. But we can help people cosmetically. And the United States Army came to see Jay, myself, and one of my former postdocs, Linda Griffith, a few years ago, and they said, we have people coming back from soldiers, coming back from Iraq and Afghanistan, said, could you ever make new body parts for them, like a new ear? So on the next slide, what Linda did is she made an ear, as you see on the top, but if you look on the bottom right, what you see is a high-powered scanning electron micrograph of polymer fibers where we've taken chondrocytes and they're growing on the scaffold, and over time you'll get pure cartilage, and the scaffolds will disappear because they're degradable. In fact, they'll break down into water and carbon dioxide. And what that means is when you put this ear with the human cells, you could put it on a rabbit. And this was actually done by the Vacantes. The next slide shows a picture of the human ear on the rabbit. And you can see that. And finally, we go to the next slide because we actually did do this on patients. Next slide shows a 12-year-old boy that my colleague Jay treated. And this 12-year-old boy, like others, likes to play baseball. But if he ever got hit in the chest with a ball, he could die because he's got no chest covering his heart. So Jay operated on him. We made a scaffold for him, and Jay took the boy's own cells and made him a new chest, and he's now 6 feet tall and doing well. Another thing we did is skin. Next slide. In this case, we actually licensed this to some companies, and here's a little boy who's very badly burned. And you can see that. Now here the product is going to be neonatal skin fibroblasts. Next slide. And you can actually cryopreserve these. But then what you can do is put them on the patient, on the little boy, at the time of the injury. Next slide. And let's come back 3 weeks later. If we could go to the next slide. Yeah, thank you. So you see he's doing better 3 weeks. Now let's go to the next slide, and we'll see him at 6 months. And as you can see, he's doing much, much better. Now, these have been approved for patients with diabetic skin ulcers and also patients who are burned, like this little boy. And they're now used by several companies, and they've treated many, many patients. I thought I'd do one last example to illustrate a slightly different point. Not only can you make new tissues and organs, but you can, and we're working on many different kinds. We're working using these principles on making new blood vessels. That's now just being done and completing phase 3 clinical trials, final clinical trials. We're working on spinal cord repair to hopefully help people walk again who are paralyzed. We're working on hearing loss, in that case actually by discovering some new molecules that may stimulate hair cells. But and we're also working on making a new pancreas for diabetics. But what I thought I'd end on is one other thing, which is that you could also use these approaches, and we are, and some of my students are, as well as other people, make organs and tissues on a chip. So here I want to tell you about the work of several of my students when they were in our lab, and they've now actually started a company to do this. So let's say you wanted to make a heart on a chip, and the next slide shows what they did. You can take what are called cardiomyocytes, and if you give them the right media and put them on the right polymer scaffold, they will have almost the exact architecture of heart tissue. You see sarcomers and an intercalated disc. And what's really striking is you can take these systems, and if you look at them under a microscope, you could take 6 million cells. And I will just show you a video on the next slide of this. And we're not shaking this or anything. But what's really amazing is here you have 6 million cells, and they're beating like once. It's like this muscle, your heart beating, and yet it's in a dish. It's not in an animal, not in a person. So you have these 6 million cells in a dish, and that's what it looks like. So what you can actually do, and I'll go to the last slide that I want to show you today, is actually take an EKG of it. So you can really, these actually really mimic exactly what happens today. So now what several of our students, Milika Radzek, who's at the University of Toronto, and Gordana Vaniak, who's a chaired professor at Columbia, they started this company, which they asked me to help a little bit on, called Tower Biosystems. And because you have this heart and a chip, what they're doing is you can screen for toxicity and see if the heart tissue, if the molecules that people are studying cause toxicity. You could also actually use it to test new drugs. Are they effective in taking heart tissue on this heart on a chip? And not only can you make a heart on a chip, others have made livers on a chip. And another one of my former students, postdocs, Gio Traverso, he's made a gastrointestinal tract on a chip. And another ones have made lungs on a chip. So this has the potential to someday minimize animal testing and I hope minimize human testing. So, but you can make all these. Finally, it's interesting. People have done things with this tissue engineering that I'd never dreamed of. Like one company I know of is making leather, and quite a few companies, including one by Shulamit Levenberg, another one of my postdocs, they're actually making meat this way, because you can actually engineer meat by, again, using the right kind of cells, the right kind of media. So it'll be interesting to see where this all goes. But already, what's been wonderful is that you can use it to make some new tissues and organs to really help people who are burned and with other medical problems, and you can develop these organs and tissues on a chip. More broadly speaking, as I close this talk, I want to thank the organizers for asking me to do this. And what I have tried to get across is how chemistry and engineering can do things that I hope are having a big impact on medicine, everything from new drug delivery systems to COVID vaccines to creating new tissues and organs. As you can see from what I went over, that I had certainly a very rocky road for early in my career where people were very skeptical of these ideas. I think if I have any things to impart to the people in the audience, not that I'm any expert, but it's to dream big dreams that you hope can have a big impact on science and medicine, and recognize that you may often have a lot of obstacles, people telling you that your ideas will never work. And I think it's really important to not give up, to keep pushing and pushing, and hopefully your big ideas and big dreams will come true. Thank you so much again for having me. It's been an honor to present this.
Thank you, Bob, for a very fascinating and inspiring lecture. We have a number of questions that have come in, and I'm going to quickly ask you a series of questions one by one. First is, let me start with one of the questions that came in. I'm going to introduce that question with my own personal experience with you over the last more than 25 years, almost 30 years. One of the most impressive things about your work and the way you manage it is time management. Even 2 days ago when we talked, I sent you an email, and within a few minutes we were on the phone together, and it was only early morning in Boston, on the east coast of the US in Florida. So the question was, how do you balance between, how do you manage your time given your multitude of interests, and also how do you manage between faculty research work that you do, mentoring students that you do on a global scale, and also your commercial research and involvement in FDA and boards of companies?
That's a very good question. I've been asked that before. And actually in, I think 2009, Nature wrote a 3-page article just where Helen Pearson, one of the editors, followed me around for the entire day. And it was educational even for me. I think, well, what I do is, well, first I have lots of short meetings, 15-minute meetings, with different people. And, and I also have I'm very lucky. I have a terrific staff at MIT, both in terms of people in the office and people in the lab, and they help me out a lot. And, and actually the other thing I've done, which probably will sound crazy, but I don't, is travel. Like I never stay any place very long. I always try to get back as quick as possible. The example I gave, which is true, is 5 years in a row I had to go to Israel for for something like a lecture or honorary degree or something. And each time I actually didn't use a hotel. I flew from Boston to New York to Tel Aviv. I gave the lecture. I had dinner with some of my students, and and then they have a 1 a.m. flight, and I just flew right back. And so I really don't, spend a lot of time smoozing, I suppose. Even when I was in Singapore last, I did pretty much the same thing. I was in Singapore for just about 24 hours, and I was able to give a lecture and have a number of meetings, have dinner with a bunch of my students, and then hop on a plane. And it was a long ride back, but but those are some of the things I do. Commercially, we maybe get involved in a company in a year. Pretty much every company I've been involved, actually, it's my students that are also involved, and I do a lot, and I have a very close relationship with them, and it's like I work through them, and I'm often on a board of directors and scientific advisory board.
Thank you, Bob. One of the questions that came from one of the young scientists is recently there have been reports of, at least the question, that's what it asks, apparently increasing cases of allergic reactions to mRNA vaccines as a consequence of polyethylene glycol. Do you have any perspectives on how to address it in the future, or is it an issue?
Yeah, well, first, I think it's speculation. There have been, I think, a few very rare incidences of allergic reactions to the vaccines, which could be for many reasons. I think there's speculation that it's polyethylene glycol. Whether that's true or not, I don't know. But there are some. But the amount of we people may not realize it, but we take lots of polyethylene glycol every day. I mean, it's actually in different things. Like, for example, if anybody's constipated, which lots of people are, you take something called Miralax, and that. You take a tablespoon of it a day, and that's probably 1,000 times more than what's ever in these little nanoparticles. So I don't know that that's true, but how you address it is important. I mean, you have to do studies to try to understand what causes it, and it could be any number of things. It could be to impurities in any of the preparations, whether it's polyethylene glycol or other things. So I think you have to step by step take that apart. But so far, it's important to realize that they're very, very rare reactions. It's happened a couple times.
Bob, one other aspect, perhaps a unique and a very impressive aspect of your work, is that you have remained a faculty member and an active scientist throughout your career without leaving academia, but at the same time, through startups and also through established major companies, you have found a way to have significant impact on industry and society, especially at the intersection of engineering and medicine. So especially for the young people in the audience, do you have some suggestions on how to balance, on the one hand, basic academic research with practical and applied research, especially in the area of medicine, and also basic research in engineering becoming very applied and useful product in biomedicine?
Yeah, well, I think the important, my advice, and actually what I did is I always put the MIT work and the basic research first. That's always been my top priority, working with the students working in the lab. But one of the things that became important to me, you work on things like some of the drug delivery work I talked about. I remember I worked on for 10 years. We published papers in Nature and Science and other places, and sadly, nobody was using it. And I realized, and that includes even the things that would lead up to the nanoparticles that are, being used to, to treat COVID vaccines today. But I realized if I didn't take the initiative of trying to move these things forward, practically, nobody else would. So, I mean, maybe, I shouldn't say nobody else would, maybe over 50 years somebody would, but it doesn't happen easily. So to me, it was important to not only do the basic research, but once that basic research was working and could be useful, it was very important to me to see it help people. And if I didn't get involved in companies, probably it wouldn't have, or it certainly would have happened a lot less.
One of the people online asked the following question. So, can you comment on the potential of bioprinting technologies? How far away are they from clinical use?
Yeah, well, I think bioprinting is very exciting, but I also think that the number one issue in terms of there's a couple of things you can use it for that are important, right? One is like an organ or a tissue on a chip, and I think that's exciting, and that could probably that could be a surrogate for animals and humans at some point. I think you'll still need to use animals or humans, but you could do high throughput screening. So that I think is already there in some cases. There are companies doing that, and I think it has accelerating some drug development, some drug toxicity. So I think that can already be done. I think in terms of really making tissues for patients, that's going to be longer. And the reason for that is understanding the biology well enough. I think we really need to understand the biology to guide what you want a 3D print. And I think that will take longer, but I think someday that'll happen as well.
Yeah. Several people asked a question. I'm going to combine all of them into the following question. We have time for only one or two more questions. So at the start of your career, like most scientists, when you're young and trying to prove an unpopular or unconventional idea, you faced a lot of skepticism from established researchers. Do you have particular advice for our young scientists who are joining us online on what kept you going? And what advice do you have? Now this is almost 45 years later. The world has changed a little bit. Should they do something differently from what you did then?
Well, I think that, there's a couple of things. I think the most important thing, I think, for young scientists is to do just great research, to dream big about really asking really big questions that, if you're successful, might have a really big impact. I think you should recognize, though, that if you do that, sometimes people are gonna, you might run into what I'll call conventional wisdom, and people won't like it if you upset the apple cart, so to speak. I'm sure you've seen that too, Subra, and in your work. And so I think when you do those kinds of things, I think you just have to hang in there. You have to believe in yourself, and you have to keep trying, and it may take a long time. But I always believe in dreaming big and not giving up. And I guess I've been very stubborn, and I think it's just important to ask big questions and then not give up easily.
Thank you. And Bob, I know for several decades how busy you were traveling. So here is a personal question to you. Have you missed travel in the last 8 to 10 months or so?
I miss. It's funny. I don't miss being on the airplanes. One of the nicest things for me traveling, like the last time I was in Singapore, I had dinner with probably about 15 of my students, and that was great. And when I mentioned Israel, I think I was there and I had dinner with about 20 of my students. So seeing people that you care about, in different places, I miss that. But I have to say, not being on an airplane, I don't miss that. And it's interesting. I look back at my life, and it's now I'm 72. And last year, 2020, is the only year I can think of where I have not been the least bit sick. I didn't get a cold. I didn't get anything. And I'm sure it's because I didn't travel. So, so I've gotten very, very used to Zoom, and, probably on Zoom 12 hours a day. So I think the answer really to your question is it depends. Some aspects I miss, a lot of aspects I don't miss.
Well, thank you, Bob. I wish you many, many more years of good health and free of any even mild flu, so that not only for you but also for the global scientific community. And on behalf of NRF and the organizers and all the people joining us online, let me thank you for taking time late in the evening, your time, to join us online and giving a very inspiring lecture. So it's also a great pleasure for me to see you and connect with you in this particular way. So thank you very much.
Well, it's an honor for me. It's wonderful to see you again, and it's wonderful to be able to speak to all the young scientists in the audience. Thank you so much for having me, and thank you, Subra, very, very much.
Thank you. Bye-bye.
Bye-bye.
Good morning, everyone. My name is Wee Joo Cheng. I'm from the Yong Lu Lin School of Medicine in the National University of Singapore. It is my pleasure to be the moderator for this first panel discussion session at this year's Global Young Scientists Summit. In this session, we will be discussing the potential use of very exciting gene editing technologies, particularly the CRISPR system, for personalized medicine. As a clinician, a hematologist that treats patients with blood disorders, particularly blood cancers, I can fully appreciate how blunt some of our treatment tools are today that we have, with a lot of non-specific activity, off-target activity, and really not addressing the key issues that underlies many of our diseases, which are genetic based, whether they are germline mutation or somatic mutation, as in the case of cancer. So, as a physician, I was particularly excited about the emergence of these technologies and what the future may hold with the use of these technologies in treating diseases. Joining us in our discussion today are two Nobel laureates. Well, in fact, this is the first time I've ever spoken to one, let alone two, in my life. So this is a real honor for me, and preeminent experts in the field. It is my honor to introduce Professor Thomas Cech from the University of Colorado Boulder from the United States of America. He was awarded the 1989 Nobel Prize for Chemistry for his discovery of the catalytic function of unprocessed RNA. He's also well known for his work with telomerase and the telomere. The discovery that he made provided some new tools for gene technology. Our second panelist is Professor Jennifer Doudna, who actually, as I found out, was Prof. Cech's graduate student. So the connection here is extremely strong indeed. And Professor Doudna is the professor in the Department of Chemistry and Department of Molecular and Cell Biology at the University of California, Berkeley. She's best known, and I think for those who are all in the call today would clearly know that she did pioneering work in CRISPR gene editing, for which she was awarded the 2020 Nobel Prize in Chemistry, along with Emmanuel Charpentier. So who better to discuss gene editing and personalized medicine with us today than both Prof. Chek and Prof. Dautner. Today, we will be discussing the following areas. We'll start off with a short description of how CRISPR works, and then we will discuss the potential medical use, what could potentially be the low-hanging fruit, the initial targets for medicine and treatment, what are the ongoing technical issues, safety concerns, and then whether there are any ethical issues that need to be addressed or not. The flow for this session would be in a form of a fireside chat. So I'll be, talking to the panelists, but we invite our participants to send in their questions through the chat function throughout the entire 1 hour of this session. And we have staff that will identify the interesting questions that we may pose to our panelists for further discussion, either during the discussion itself or towards the end of the session. So do not wait till the end to send in your question. Do send it in as and when you have question and the discussion is going on. We also hope that the session will be a bit more interactive. So for those questions that are selected, we will try to let the questioner come online to ask the question personally. So we'll, our staff will actually open up the video and the voice for you to ask the question. So without much further ado, we'll commence the session. So good evening, Jennifer and Tom. Thank you for joining us for this session. I think maybe we'll start with a more general question on, what is CRISPR? How does it really work? And why is it potentially better than previous gene editing technology, say, for example, like TALENs and so on? I think maybe I'll address that to start with to Jennifer to kick us off. Thank you.
Well, thank you, Weiju. And I just want to say good morning to everyone in Singapore. It's really an honor for me to be here, especially with both of you and with Tom Chek to discuss this. And so CRISPR is in biology, it's a bacterial immune system. It's a way that bacteria find and destroy viruses, and they do it using an RNA-guided mechanism. And the reason everybody's really excited about it is that it's become a common technology now for genome editing, using it to change DNA sequences in cells in a precise way. And so it provides the kind of precision that people have been wanting for manipulating genomes. And it provides that precision in a very accessible, simple to use kind of package that makes it easier to use than earlier genome editing technologies. So that's really what's happened over the last 8 years, is that it's just sort of taken off as a tool that lots of people now are using globally, really, to do gene manipulation in plants and animals and human cells.
Thanks very much, Jennifer. Tom, you want to add anything to that?
I would love to add maybe a couple of things. Just in terms of the technology, I find it important to realize that the RNA-guided CRISPR system does the cutting of the DNA. But then what happens afterwards is done by the human cells repair machinery. And so we have a little less control maybe over what the human cell does than we did with the cutting itself. So that may come up again in the discussion. The other thing that I think we could mention at the very beginning is that my laboratory and Jennifer's laboratory and many other laboratories using this as a tool in the laboratory with human cells, it's very they're very accessible when they're growing on a petri dish, right? You have good access to the cells. So even though this is a fantastically robust machinery, getting the delivery issue for human disease is going to be easier in some cases and very daunting if you need to deliver it to the middle of the brain, for example. So maybe the blood system, the hematopoietic system, will be one of the most accessible tissues in the human body. So those are just a couple of additions to Jennifer's nice introduction.
Great. Thank you very much. And as you mentioned, you have started to mention about, the potential application in medicine. Perhaps I would, get your view as well on what do you think are the real kind of medical potential? Is it mainly to deal with genetic disease, or there are sort of broader application beyond that to start with? Maybe Jennifer, you can start with that again.
Well, genetic disease is clearly one exciting application of CRISPR, and as Tom said, using it in the blood supply, in the blood system, to change the genes that are responsible for causing things like sickle cell disease. That's already underway, and other blood disorders is one of the early applications of CRISPR. And then there's, of course, many other opportunities to correct disease-causing mutations if the challenge of delivery can be addressed, again, as Tom said. But I would also point out that one of the things that's really interesting about CRISPR is that it goes well beyond human medicine. So it's also being widely used now in agriculture. It's actually being used widely in microbial systems, microbiomes, and doing microbial editing there. And actually, in the face of the current pandemic, there's a very active effort in a number of labs and companies to use it as a diagnostic tool as well. So there's, it's a very versatile kind of system that can be deployed, and it's all kind of fundamentally dependent on the RNA-guided mechanism, which makes me and Tom very happy, I'm sure, because we're both kind of RNA lovers from way back.
Absolutely.
Yep. Tom, do you have anything to add to that?
No, not really. I mean, I think that one, again, I'm not nearly as much an expert on this as Jennifer is, but one disease state that am very excited about, that's just a terrible disease, is muscular dystrophy, which is a genetic disease, and the children usually die when they're teenagers. I knew one who passed away when she was like 18, and this normally male disease, but this is an area where Eric Olson at UT Southwestern in Texas in the United States has made some just remarkable progress, interestingly, by changing the RNA splicing pattern of this large gene. So I think that human ingenuity and scientific, creativity are so powerful that we will find ways to overcome some of these challenges. And the fact that we're starting out, due to the discovery of Jennifer Doudna and Emmanuel Charpentier, with such a robust tool that has such high activity, really makes it worth trying to make a medical miracle out of some of these ideas.
And maybe as a non-expert, although I do work in the cancer field, and we do know that, say in cancer, when we talk about genetic defects, it's actually a whole range of different kinds of abnormalities. From sequence mutation to translocation to deletions and so on. Is this technology potentially applicable to correct and reverse a whole spectrum of different abnormalities, including, say, the creation of new super enhancers? Say, for example, can we actually delete the super enhancer and hence reduce the overexpression of certain gene? Is it as broad as that, or actually its main application is towards certain types of genetic abnormalities and correction of that only? Jennifer, maybe? Well,
it certainly can be used for that kind of manipulation. I think in the cancer field, and I'm curious about what Tom thinks about this, but my view of that right now is that probably CRISPR will be useful primarily as a way of manipulating the immune system, immune cells, to get them to attack cancer. I think it's probably going to be very challenging to use CRISPR directly in tumor cells, because you'd have to get every one, and I think that's going to be hard.
That was exactly what I was going to say, that you could think, okay, well, you've got a mutant KRAS, which is driving cancer. We know that CRISPR can be used very efficiently to remove a gene, okay? But now the problem is let's say you get it up to some remarkable 90% efficiency. Well, that may be of very little value, because the other 10% of the cells will quickly take over the population. So there are some limitations that are just part of the biology. The biology gives some limitations.
Mm-hmm.
Yeah, great. I think maybe we can move on to the next issue, which we have started to touch on, actually, which is what are some of the technological hurdles and challenges. And for this, we have some questions from the audience, which I think are already immediately relevant. We have started to talk about the use of CRISPR in complex genetic diseases like maybe cancer. And I think there is a question from Christopher Ortiza. Can we get him up online next? About the use of CRISPR in, say, germline genetic conditions that may be driven by 2 genes. So, can you target more than a single gene at the same time? But I'll get him to come online and address the question. Are we able to do that? Christopher, if you are online, could you click on share? And we will be able to get you up on the screen to ask your question. Great. I think he's joining us soon. The technology is working, hopefully.
I'm impressed.
Yes. Hi, Christopher. Very good morning to you. You have the airtime to ask your question to the 2 experts. Go ahead, please.
Thank you. It's an honor to see 2 Nobel Prize winners in chemistry in one screen. And actually, the question I want to ask is, how are we going to deal with genetic diseases that are associated with multiple genes, not just by a single gene, using CRISPR?
Yeah, well, I can take a stab at that, I guess. And I guess the short answer is it'll be harder, because it becomes a kind of a combinatorial problem where you want to target multiple genes, and if you need to have editing occurring at those multiple genes in every cell, then you can imagine that it becomes more challenging, certainly, than if you're going after a single edit. That being said, there's some really interesting, I think, and very encouraging work that's being done both in animal cells, maybe in some human cells, but certainly in pigs, in mice, where multiple edits are being introduced quite efficiently. And also in plants, amazingly. In plants, there's a lot of work, and people refer to it as multiplexed editing, where you can use a series of guide RNAs that direct the Cas9 protein to different genes at once in the cell and to induce editing. So I think it's sort of at what I would call the kind of the bleeding edge of the technology. It's a direction that a lot of labs are working to improve efficiencies, but still needs further development.
Okay. Thank you, Professor Dodna.
Thanks, Christopher. So, we're moving into the area of technological hurdles, I guess. And actually, Tom had earlier mentioned that in his, well, at least the first thing that he mentioned in his mind was potential issue with delivery of CRISPR to, especially in the human system where it's so complex and there's a lot of hurdles in the way to getting somewhere to a target site. We have a question from our audience in the relevant area. Philip Boscovich, we're trying to get him online to ask his question, and in fact he has two questions. Okay, and he's coming in now, soon. Okay.
Tom, you should take this one.
Sure.
Yeah. Philip, good morning to you. Thanks for joining us. I think it's good evening to you actually on your side, or good time. It looks very late on your side. Please go ahead and ask your question.
Yeah, Cambridge now is night. So the question that I was keen on, and I think the one that links also to the Professor Langer talk, can we actually make CRISPR-Cas systems smaller, or can we actually make gene editing systems smaller so it's easier to be delivered? And I think that's one of the key problems, because Cas9 is huge, complex machinery. But can we somehow, by engineering it, now with biotechnology and the chemistry, can we make it simpler? Yeah. Thank you.
Yes, thank you, Philip. I think that's certainly something that progress can be made. One way to get the Cas9 into a cell, of course, is to put in the gene for it, so that the gene would then be expressed to make the protein. Since you have to put in the guide RNA anyway, which is a nucleic acid, one might be able to co-package the guide RNA with a gene for the Cas9. It's just one of many ideas. I'm not trying, I have no patent in this area. I'm not trying to sell anything here, but that's one possibility. I might just say that the recent success of the Moderna and BioNTech vaccines that are fairly large messenger RNAs being delivered by lipid nanoparticles gives me some hope, that delivering nucleic acids, which has just been a very tough road for many decades, that there are some new breakthroughs in the engineering of these lipid nanoparticles. And, I'm pretty optimistic.
Philip, I think you have a second question. You want to ask that as well about using,
Yeah, I think that might be relevant to the previous, but CRISPR-Cas look, at least for me, as you mentioned, not only for monogenetic diseases, but can we actually, for tandem repeat associated disorders, actually solve that just by actually removing extra DNA?
Like trinucleotide repeats?
Yeah.
Jennifer, you want to take that one?
Sure. Yeah. So I think that's a really interesting question. And in our own lab, one of the very first applications we had for CRISPR in my my academic lab was to target the gene for Huntington. That, as you may know, when it has an extension of triplet repeats, that actually induces disease, Huntington's disease. And so what the strategy that was taken by a former postdoc, Brett Stahl, who was doing this work, was to look for single nucleotide polymorphisms that you could actually target on either end of the repetitive, sort of the large repeat element, to essentially excise it from the gene. And he, had some success doing that in a mouse model of Huntington. So that's the kind of strategy that one could take with that kind of disorder, where you could, and kind of relevant to the previous question about making multiple edits or multiple cuts in the genome. You can actually use it to literally, chop out a segment of DNA and remove it from the genome.
Yes, I like your question, Philip, because in general, it's easier with CRISPR to cut something out than to have to repair. It's more efficient to cut something out than it is to have to repair, like a point mutation. And so I think that your idea of using this with trinucleotide repeats or ALS has the C9orf72 hexanucleotide repeat. Pretty, you know, that's pretty promising area. Maybe you should work on it.
Great. Thank you very much, Philip, for your question. Thanks for joining us. I've got another question from City Oslan, which I think it's an interesting technical question, maybe helpful to many working in the field at a more junior stage, is that it's the choice of technology, really. I mean, if we have one particular gene to edit from a yeast genome, of course, this is in yeast and not in human disease, but I think it's nevertheless relevant. Based on your experience, which method will be faster and more efficient, using homologous recombination or CRISPR? And in this aspect, do we have, I guess, what is the appetite for FDA and others for doing trials using CRISPR technology in terms of funding and regulatory approval? So two slightly different questions, but I'm asking on behalf of City, who is unable to join us online. Maybe Jennifer, again, down for you. You can go with a technical question, and maybe I'll ask Thomas to address the funding and the regulatory appetite question, if you may. Yep.
So I think the technical question referred to using it in yeast. Is that correct? Yeah. And so for those that have worked with yeast, and for me it's been a long time, but I did once do a few experiments in yeast way back when I was a graduate student. But you know, yeast are remarkably good at doing homologous recombination on their own, even without CRISPR. And with CRISPR, it can be, you know, maybe even more efficient, but I think it's, you know, it's an organism that's already kind of predisposed to do that kind of genetic manipulation. So I don't know which is better, you know, for a particular experiment that you might be doing. I think in general, you might find that CRISPR makes it even more efficient than a process that is already relatively, you know, facile in yeast. That would be my answer there. But I would defer to, you know, Tom, you may you may think differently. I don't know. You have more experience, I guess, in working in yeast.
No, no, I totally agree with you, Jennifer. I was amazed when I found out that people were using CRISPR in yeast, because as the student very nicely pointed out, you have yeast is one of the few eukaryotic systems where you had this ability to do gene editing through this very facile homologous recombination system. So I think if in your lab, if you're in a yeast lab and you're totally conversant and successful with traditional yeast molecular genetics, I don't think you need to use CRISPR.
Great. And Tom, do you have any thoughts about the current appetite for CRISPR in terms of trials and so on? And is the FDA kind of likely to approve these technology?
I would like to defer to Jennifer, because she's thought about 100 times more about this than I have.
Sure thing.
Well, you know, I think I have a couple of thoughts about that. First of all, I think there's a lot of interest on the part of regulatory committees like the FDA to figure out how they're going to appropriately regulate a gene editing tool like CRISPR. And, you know, because we're talking about personalized medicine here, I think it's appropriate, you know, it would be worth mentioning that on the one hand, CRISPR is great as a personal tool, because the whole, you know, the beauty of CRISPR is that it's programmable. So you can literally personalize it to a particular genome, which is great. But the flip side of that is that, you know, taken to an extreme, it would mean that, you know, CRISPR for each of us would be different. And so how would you ever do clinical trials where you are testing something in an individual that then has to be changed in somebody else? So that's clearly not going to, you know, that won't be possible to do a clinical trial for every kind of iteration of CRISPR. So I think one thing that's happening right now is looking for diseases where there's a way to make CRISPR as a tool that is more generally applicable. So, and a great example is going back to what we said earlier for sickle cell disease, where it's possible many people have a similar mutation, And so you can use the same form of CRISPR in principle to treat different people. And so there you could imagine doing trials, and in fact they're ongoing. But it gets harder when we think about really rare diseases where, on the one hand, it's great to have a personal tool, but then how do you think about testing it appropriately?
Very, very fair point. Actually, I think that's always the challenge for any sort of personalized medicine approach is, how do you assess it? We have a next person who's going to ask the question live. Emmanuel Galang. He has an interesting question, which I must admit that I'm not really familiar with. So, Emmanuel, you're live with us. Go ahead with your question.
Hi, guys. Greetings from the University of the Philippines, Los Baños. It's my pleasure and my honor to be speaking on behalf of my university here. I have a question with the recent discoveries on detector, which is intended to detect specific sequences. Were there recent applications, or have there been studies which have established the tandem use of detector as well as CRISPR to address a specific medical condition?
Okay, well, I can take a stab at that. So I think with detector, you're referring to a form of CRISPR that's being used for a diagnostic.
Yes.
Yeah, and so it is CRISPR. It's just using it in a fashion where it's used for detection rather than for genome editing. And so one of the really nice things about CRISPR as a diagnostic is that it can be programmed with multiple guide RNAs to look for different sequences in one reaction. So let's take the example of the coronavirus, the SARS-CoV-2 virus, where people are discovering mutated forms of the virus that are cropping up and can sometimes be missed in standard PCR type assays, where a tool like CRISPR, again in principle, could be used to capture all the sequences that represent different forms of the SARS-CoV-2 virus because of this ability to do detection of multiple sequences at once.
And if a new variant shows up, you can add that to your list in a week, and you can have it up and going again.
Yeah.
Thank you very much, you guys. That sounds very promising, specifically with the COVID-19 pandemic that we have. Thank you so much from the University of the Philippines.
Thank you for your question.
Thank you. Great. Thanks very much, Manuel. We have rather a lot of people interested in this kind of area. So we have I guess we'll take the last question in this segment before we move on. And this is from Mark Carrascal, who is also going to ask the question online. So hopefully Mark will pop on soon. There you go. Yeah, Mark. Welcome, Mark. Thank you for joining us. You're live now.
Hi.
Please go ahead with your question to the professor.
Yes, can you hear me?
Yes, very well.
Yes. Hello, everyone. Well, first of all, I'm Mark from the Philippines. I'm actually a clinical microbiologist, and just before asking my question, I would like to, state that I'm very happy to be part of this session, especially with my I'm looking up to. So my question is really more on the practical side, because as a clinical microbiologist, we're really into that tenet that these technologies should be accessible to the people who this technology should really serve. So my question is on what are the current strategies in making CRISPR technology a more point-of-care type of technology in terms of diagnostics? And second, what are the current strategies in making CRISPR more accessible to the scientific community? Because speaking from a country with no access to this high-end technology as of the moment, we are really relying on these strategies on making CRISPR a more accessible technology to everyone. Thank you very much.
Well, I guess both of you probably could address that question from your own perspective, right? Yep. Maybe Tom can go ahead.
Maybe Jennifer could handle the diagnostic point of care one, and then I'll do the more accessible to the scientific community.
Sure. Well, the point of care idea is being developed by a number of groups actually right now who are using CRISPR to detect viral RNA in samples like saliva and nasal swabs. Making it into a true point of care test, I can tell you from personal experience, is challenging, not so much because of CRISPR itself, but it has more to do with just, you know, creating a test that's really easy to use, fast, kind of bulletproof in terms of, you know, being very, very easy. You don't need special training or instructions to do it. And where we can, you know, one challenge we have right now, honestly, at Berkeley is just dealing with our environmental health and safety people who don't want, you know, saliva or nasal swab samples that could have virus in them going into a point of care test. So, you know, we're trying to figure out how to manage that. But I think in principle, it's a solvable problem. And whether we'll see a really robust point of care test in the next few months, I think it's possible. But I'm really sort of thinking down the line about, you know, future pandemic preparedness. And as Tom said earlier, you know, one of the things that's great about CRISPR is that if a new virus pops up or a new variant of the current virus, you know, we can quite quickly create a format for CRISPR that will detect it. So I think that's the real utility there. And once you know, a format for, you know, dealing with all these other, you know, sort of logistical issues that can be solved, the CRISPR technology fundamentally is going to be very useful for point of care testing.
So Mark, thank you for your question about the accessibility of the research technology to the scientific community. We understand very well that we are in a privileged situation in the United States, in Singapore, and in many other countries in terms of the resources we have to do science, and we are sorry that those are not more worldwide, broadly available. I will say that the CRISPR technology is not particularly expensive or inaccessible. Any laboratory in your country that has the ability to grow human cells on petri dishes and to transfect plasmids into them already, this is no added expense. Of course, if you don't have those fundamental tools available, then that's a huge barrier to entry to the field. I will just remember when 6 or 7 years ago, when I was first interested in using this in our lab, I called up Jennifer and I said, we need to collaborate with you, we need some help. And she paused for a minute and she said, well, I would be glad to help, but it's really easy. She said, just try it, it will work. And so I had a student from China who was the first person at our university to get it to work, and she was able to get it to work quite quickly. So it is, there are some barriers to entry for third world countries and other less developed countries, but certainly this is not an especially high standard or especially expensive.
Can I just add one thing to that? So Mark, I'm curious whether you have access to adgene.org. Do you know this organization, Adgene?
Yes, I think I've heard of it, and some of my colleagues here, yeah.
Yeah, so it's a nonprofit organization set up in Massachusetts that distributes research reagents, including CRISPR, just at cost. So they don't, you know, it's pretty inexpensive to get it. So that would be one way. And obviously you can always reach out to us if there are, you know, constructs that you can't get a hold of some other way. We're always happy to send them out.
Jennifer, how many Cas9 plasmids has Adgene sent out around the world?
Oh my gosh, I've lost track. I mean, if you add up all the different labs that are, you know, placing constructs there and then Adgene distributing them, it's got to be, I don't know, many tens of thousands.
Yeah, I think it might be 100,000 or something like that.
Yeah, I wouldn't be surprised. Yeah.
Great, thanks.
Thank you very much. That's very wonderful. Yes, thank you very much.
Yeah. Bye-bye. I think that was a great session, actually. There, we'll move on now to some questions related to safety. There were a couple of them somewhat related, but anyway, I think it would be great if we can get the participants to ask those questions online. The first will be, I won't even try to pronounce his or her name, right? But Yosipol, who we are trying to get online next. She should. There you go. Oh, hi. Good morning, good evening to you. Please go ahead and ask your question.
Hi, Jennifer and Thomas. My great pleasure to talk to you today. I have a short question regarding a clinical trial. And I think Jennifer trying to raise the campaign of a global pause. For some example of disease like sickle cell, that it's about the disorder in genetic information. So it's only way to treat these diseases by CRISPR-Cas at the moment. But now it seems that we have the global pause to stop a clinical trial in humans. So my question is, what would you like to prove for avoiding unintended consequence of the CRISPR in humans? I mean, for example, like sickle cell, we don't have any chance to treat those people instead of CRISPR, but now we stop this trial. That means a lot of people now are suffering instead of we can cure them immediately. So what would you like to prove to make these global issues move forward?
Jennifer, you want me to say something first, and then you can add
sure.
in? Well, I think that you know that you have to go through safety tests. You usually would try this first in a mouse model of the disease, and then perhaps what in the example I gave previously, there was which is muscular dystrophy, which is a genetic disease. There turned out to be a dog model in the UK, and the researchers were able to cure this muscular dystrophy in these dogs. And so then at the same time, they're looking for side effects, they're looking for safety issues. And I think that you have to go through those steps. You can't just start with humans, because it's too risky. So I appreciate that you would like to speed this up, and we all would like to speed it up. And we can speed it up to a certain extent, but ultimately you don't want to, if you do something that injures many people, then it sets the whole time course of the thing back it gets a very bad reputation then. So safety has to be considered at every step. And usually the animal models, although not a guarantee of safety, are a good intermediate step. Maybe Jennifer has other ideas.
Right, yeah. I think, I think about this a lot because I hear what you're saying, and, people are suffering, and you want to, I think all of us as scientists, we want to see our work benefiting people as quickly as possible. But as Tom said, it's almost, it is worse to have something go forward prematurely and then harm people. So, and maybe, harm the whole field. And, Tom and I both remember, years ago when gene therapy was just coming to the fore, and there was a really unfortunate event in a clinical trial in the US that led to someone's death. And that really set back the whole field of gene therapy for at least a decade. Mm-hmm. So, I think we want to avoid that, obviously, here. But you're right. It's always a struggle, to figure out, you want things to move quickly, but not too quickly. And I think that it's just essential that, everyone conducting these trials just continue to be really vigilant about safety, and that people like me and many others who are doing more fundamental work are continuing to, make sure that the technology is working correctly, that it's not, it's not creating off-target edits, that it's, that it's ensuring the kind of changes that are going to be beneficial. And then, it's up to clinicians then to make sure that that's, when it actually gets introduced into people, that it's done in a safe manner.
Yep. Thank you very much.
Right. Thank you, Dr. Paul, for your question.
Thank you.
We have another question which I thought is quite relevant from Amit Ghosh. Again, he's going to join us online to ask his question. And this relate to, because we said that medicine, even targeted therapy, may have off-target effect, and this is related to whether CRISPR may also have off-target effect and how specific the editing is in human. Is Amit joining us? Okay, he's coming on soon. Great. Hi, Amit. Good morning.
Good morning.
Go ahead with your question to the professors.
So it's pretty straightforward. First of all, it's an honor to ask questions in this session, and very nice to meet you all. And so my question is what are the health issues which are associated with CRISPR? What are its risks? So can it accidentally shut down something important, or even cut down something in the untargeted tissue rather than the place we intended to go with in the first place?
Tom, do you want to speak to that?
Well, I'll just briefly say, and then Jennifer can add on, off-target cutting, off-target editing are unavoidable at some level. You can minimize them. Ultimately, though, there is that possibility. You'd be pretty unlucky if the off-target event shut down an important gene, but it could happen. And ultimately, that's why we have to do clinical trials, right, to see what the safety of something is. And I work for sometimes for Merck, the pharmaceutical company, and they say the best drug to take is no drug at all, right? Because every drug has some negative side effects in some people. And so of course, it's always a question of risk-benefit ratio. If you're going to die from a disease, if it's deadly, then this risk of an off-target effect is probably something you would dismiss rather quickly. On the other hand, if it's just sort of an annoying disease that you have that can be treated by other means, then it might not be tolerable to do CRISPR editing. Jennifer, you want to add to that?
Yeah, I know. You said it very well. I agree completely. I think it's all about, the risk-benefit ratio, and for severe disease, you're going to be more willing to accept a risk than not. I would just add that, I think, and you said this already, Tom, that, there's probably never going to be a therapy of any kind that will have zero risk. Yeah. So, we're always going to be dealing with some level of risk. And with CRISPR, one of those risks is some kind of off-target editing. And so, we work on ways to reduce that and, and to try to get it to a point where it's really considered safe enough, given the benefits, to use it there as a therapy.
Yeah, great.
Thanks very much, Amit. Thank you.
Thank you so much.
Thank you, Amit.
To a next question, which I thought also is quite relevant and interesting. We've focused a lot on the genome so far in our discussion, but this question next from Maya from the University of Auckland. He's going to ask the question online, is related to, whether, CRISPR could actually induce certain autoimmunity or some immune reaction that may be potentially harmful to the patient that's being treated. So let's get Maya on to ask the question. Great. Ooh. Maya, can you turn on your video?
Maya is very shy.
Well, he looks good, though. He should show his face. If that was indeed his photo. You're on mute as well. I think, I'm not sure. Is he on mute?
Hi, can you hear me?
Okay. Yeah, Maya, why don't you go ahead and ask your question, please?
Sure. Hi. It's an honor to be here in this panel discussion. Exciting topics. So yeah, my question was more about immunity. I'm not from this background. I don't have a strong background in CRISPR and genome editing, but I'm quite interested to know whether for potential CRISPR treatments for genetic disorders, is there any concern regarding any immune responses, against CRISPR? Any thought about that?
Yes, I think so people are worried about that with the mRNA vaccines right now against coronavirus, right? So putting in foreign RNAs into the human body, because there is an innate immune system that we have that is poised to be set off by RNA, because RNA usually means virus coming in, right? So from the outside. So it is a concern, and maybe the Moderna and BioNTech vaccines will give us some idea about whether we need to worry about the RNA component of CRISPR.
And actually, I thought you were going to say, Tom, that it seems like another angle to that is the question about natural proteins that are made in cells. So let's take the example of muscular dystrophy that you brought up before. You could imagine that for a patient who is affected by muscular dystrophy, where their cells are producing one form of this dystrophin protein, when you edit that to make a kind of a corrected form, that might look like a foreign protein. And so could that induce an immune response? And I know that's something that people like Eric Olson, who are working in the field, are thinking about.
So, Mayar, I think the answer to your question is we always worry.
Yes. Right.
Great. Thank you so much. Thank you so much for your time.
Yeah, thanks, Maya. I think a lot of the questions in this segment relating to safety really have to be sort of observed and pan out in the human trials, right? I mean, a lot of these are theoretical in nature at the present time. So I think the next phase of CRISPR, as we do more clinical studies, will definitely be able to learn more about that. I think we are doing quite nicely on time to move on to the very last segment, and we already have lined up 3 questions from the audience regarding ethical aspects of CRISPR. I know this sometimes can be a bit uncomfortable, but I think it is nevertheless something that is on top of people's mind. So the first person that we will have asked the question live is John Guzman. Yeah, he's joining us online now. John, please go ahead with your question.
Wait a minute, I'm having some problems.
No, we can hear you well.
We can hear you.
Oh, wait a minute. Can you hear me?
Okay, we can't see you. We can hear you, and we see a nice photo of you. I presume that's how you look like.
All right. So it's actually, first of all, I would like to thank everyone. Thank you for the opportunity of asking the question. Actually, I'm going to ask this question in behalf of some of my students. And I'd like to ask, so since we're talking about personalized medicine, how do we address the worries of the people regarding this technology being used in quote unquote playing God, since there are issues concerning the development of what they call the designer organisms, especially now that biohacking or the DIY or the citizen or the DIY science is on the rise?
I would say that Jennifer Doudna has been a real leader internationally in thinking about the ethics of the CRISPR genome editing. So maybe she would like to start on this one.
Well, it's a great question, John, and thank you for asking. I think it's something that all of us working in the CRISPR field think about, because it is quite a profound thing to have a technology that allows precise alteration of a genome in any organism, but including in humans. And if it were to be utilized in a human embryo, that creates a heritable change that makes a change that can be transmitted to future generations. So it really is a technology that requires, I think, careful thought, careful regulation. My view of it right now is that I think the benefits of using it to treat diseases in individuals is so exciting and so profound that we certainly want to see that field moving forward as quickly as possible. And it looks like we might have lost John. I hope he's still able to listen. I think that, and I think many people in the international scientific community agree that that it's not an appropriate technology to use right now in human embryos for many reasons. So the vast majority of work that's going on around the world, both in academic labs and in companies. Oh, there he is. Hi, John.
Sorry.
Is really focused on treating disease in an individual, not to create a heritable change. And there, I would argue that CRISPR is, then very much like any other type of therapy that you might utilize. It affects a person, and you want to be sure it's safe and effective in that individual, but it doesn't affect anybody else.
All right. Thank you. Thank you very much for that. Let me just share with you that actually this question, when I reported or when I taught my students about this CRISPR, especially in your recently awarded Nobel Prize, they kept on asking that if we allow these people, are we trying to engage them in doing science, or do we have this sort of gatekeeping in allowing people with no, or how do we say this, formalized training in science in doing this CRISPR? Yeah. So
yeah.
Thank you very much for that.
Thank you, John.
All right.
We have another question from Yusang Dorji, I think. Again, on the area of ethics. I will ask the question on her behalf because we couldn't get a response from her. She is coming on. Oh, great. Well done. And I think this is about, again, similar to the previous question, the issue of how it may affect not just the human, but other species as well. Yusang, please go ahead with your question.
Hello, everyone. First of all, thank you so much for this amazing opportunity to talk in front of 2 Nobel Prize winners. So my question was related to the CRISPR technology. This provides a lot of opportunities, but there are also some ethical concerns, not only for humans, but it will also affect other species. So are there any ways that we can address these issues, ethical issues related to CRISPR?
She disappeared. Well, thank you, Yusang, for that question. And I agree. I think, again, we have to be very thoughtful about how a technology like CRISPR is used in the environment. One example of that we haven't discussed yet in this session, but is certainly a topic that comes up frequently in the CRISPR community, is using CRISPR to create changes that can be spread quickly through a population, for example, in mosquitoes. And we call that a gene drive. And, in principle, that is a use of CRISPR that could, cause unexpected or unintended environmental impacts if it, kind of got sort of out of control. On the other hand, it could have a very positive impact on human health and sort of public health if it were possible to use it to control a disease that's spread by mosquitoes. So, again, one has to kind of balance, risk and benefit in that type of application. And again, I have to say that at least right now, I've been quite encouraged by the response globally of the scientific community. I think people have really recognized that this is not a tool to be deployed, without caution. So there are a lot of groups that are working in these areas who are, doing research, but doing it in a way that really takes into account the kind of safety measures that we need to have to ensure that we don't see unintended consequences of CRISPR.
Tom, you want to add anything to that?
No, I thought that was a very good response. Thank you, Jennifer.
Cool. Great. So we'll move on to the next question from Eric Onos. Again, in this general area of ethical concern, Eric will join us. Eric, please go ahead with your question.
Hello, everyone. I'm Eric from University of the Philippines. First of all, I'm very thankful for this opportunity. So, in 2019, Professor He Jiankui from China was criticized for his controversial research on twin babies, CRISPR babies, in whom he tried to edit the CCR5 gene for, hopefully, for HIV prevention. So there are legal and bioethical concerns that were raised by this research. Can I ask the panelists to comment on this issue? Also, do we have global efforts to prevent this controversial research? And ultimately, do we think that CRISPR technology can be ultimately used for editing out genomes in human embryos? Thank you.
Well, I'm actually curious, so I said a few things about that in response to a previous question, but I'm actually curious about Tom's opinion about this, because what I've kind of sought to do, especially with respect to the He Jiankui situation and possible future use of CRISPR in human embryos, has been to really try to engage the international scientific community through the scientific societies and through other organizations that have weighed in on kind of the appropriate ways to proceed with the technology. And I'm curious, Tom, whether you feel that is an effective strategy. Do you think there are other things that could or should be doing to address this issue?
Not really. I think that again, the distinction that is important here is between germline editing, which would be inherited by subsequent generations, and treatment of somatic tissue or stem cells within an individual, which would not be inherited. And so it makes total sense to start with the somatic treatment and to treat the individual and not the germline, knowing, of course, that if it's a serious genetic disease, what you really would like to do would be to eliminate it from the germline. But it's sort of a stepwise thing, and there are so many possible misuses of the CRISPR in the germline editing for enhancement of various traits rather than curing diseases. I don't know if I should say this in public, but I think this will come, at some point, the germline editing. I think it's just the history of science that if something can help people, you can't keep it bottled up for forever. But I think that for the coming, decade, maybe more than that we should avoid, we should have a prohibition on those experiments until we understand more about safety and efficacy.
Right. Yeah.
Thank you so much.
Thanks very much, Eric. Thank you. I think we're doing fantastic for time. We have a couple more minutes. We have some fantastic question. I think it has gone better than I expected. The technology actually did work very well for us. So maybe I have, I will ask a couple of, in fact, related question. Just one last question that in screening through the questions, there were a couple of colleagues that have asked this question. And I'll ask this question of Tom, because he's, as he says, an enthusiast for the CRISPR technology. And so he should be unbiased in that sense that he can look at the CRISPR in comparison to other technology in an unbiased manner. So there were a few questioners that asked about what are the, with the advent of CRISPR, are there still useful roles for other technology like TALENs, zinc finger nucleases, Cre recombinases, or even the RNAi, short hairpins, the kind of technology in science, or everyone should be just using CRISPR because it's so much better, more accurate, cheap. There's no reason why you shouldn't use it. So Tom, what is your take on that? Do you use other technology in your lab still, or are you just using CRISPR?
Well, I think that I may disappoint some of my friends who invented some of these other technologies, but I think that the zinc finger nucleases and the talons are history at this point. They are so tedious and expensive and less efficient than the CRISPR. I think they're sort of gone. Maybe that's too extreme. The microRNAs, short hairpin RNAs, I think have, we still use them. I must say that they almost are guaranteed to have off-target effects, often quite serious ones, which people don't really think about. They don't want to think about them. So I'm afraid that that is a bit of a primitive technology, in my opinion. And I mean, after all, biologically, think about it. The microRNAs are meant to control a whole pathway, a whole series of genes. So having this sort of broad specificity rather than precise specificity is critical to their biology. So I think that they're somewhat on the way out as well. Although they will still be used. The Cre recombinase, we actually use that in conjunction with CRISPR sometimes. So it's a tool that has some use for recombining out a segment of DNA in the genome. Although again, it's not without its attendant problems.
Great, thanks very much. Jennifer, anything to add to that?
Well, I guess just real quick, I would add that you know, I think that you know, we use RNAi methods in my lab now still, you know, but for particular purposes. Like if we're really interested in not manipulating the gene itself, but manipulating the RNA transcript of the gene for some reason, then you know, that is the way to do it. So I think you just have to look at, you know, what it is you're trying to do, and then use the tool that makes sense to do it.
Yep, great. Thank you very much. Thank you both for your excellent, you know, answers to all the and patience actually with the technology, and all our participants with your question and addressing these very important issues with us today. We've come to the end of the session. Enjoy the rest of the meeting and hope to see you all soon. Thank you very much.
Thank you, Wiju.
Thank you, everybody. Thank you, Wiju. Thank you, Tom.
Thank you, students from all around the world.
And students, yes. Thank you.